It's really a pleasure to be here, and I will tell you in few words what we are focusing in my lab. So we switch from cancer mainly to brain, as Yael mentioned. The main focus of my laboratory is we are trying to understand the main mechanisms that underlie memory function, and particularly brain plasticity in very important region in the brain, hippocampus. This region is really involved in learning and memory functions. And one of the major questions that we would like to address are what are the mechanisms that maintain plasticity of hippocampus? And what are the mechanisms that initiate experience-dependent gradual transition from physiology to pathology? As most of you know, Alzheimer's disease is the most frequent and common form of dementia. And maybe you hear that in the last 10 years, unfortunately, all the clinical trials fail. And great minds now trying to understand why it's happened. And if you are looking literature during the last 50 years, it's becoming clear that hallmarks of Alzheimer's disease pathology, which most of you know, the plaques and tangles in the brain, are not correlated to decline in cognitive function. So many years, people try to design drugs to reduce these plaques in the brain, but it did not work out. And if you're looking current literature, it's become clear that dysfunction of synapses, it's a contact between neurons in the brain, could be really the primary cause of the disease. And what it's clear now that there is a really very good correlation between cognitive decline in Alzheimer's patients and reduction in synapse number. And what we're trying to understand is what is the primary cause that initiates the synaptic loss. Because obviously all of you understand that we would like to design the drug that can interfere with the stage that can be reversible, right? In Alzheimer's, a lot of neurons die. And many biologists, and me too, we think that at this stage it's irreversible. But if we can design a drug and we can understand the process of reversing synaptic loss, we still believe that it could be really functional for Alzheimer's disease pathology and neurodegeneration. I would like briefly to explain you what is the current state of knowledge in Alzheimer's disease. So as you can see, Alzheimer's disease is featured, as I told you, by plaques, which are composed by amyloid beta peptide. And this peptide is produced by proteolytic cleavage of amyloid precursor protein, which is a transmembrane protein in the brain. And I would like to mention that each of us, doesn't matter what is our cognitive state, has this precursor protein, and in each of us brain, we have this amyloid beta peptide that can be produced by different lenses. Short, for example, amyloid beta 40 amino acids, or long, like 42 amino acids. And during the last 20 years, many familial Alzheimer's disease mutations have been discovered that increase production of this toxic amyloid beta 42 isoform, which has high tendency to aggregate. So what we know today is that amyloid beta peptide can be in monomeric form and can switch to oligomeric and even fibrilic form, and this process is dependent on the concentration of the peptide. The more peptide we have, the faster the kinetics of aggregation, and on the ratio of these two isoforms, amyloid beta 14 and amyloid beta 42. So the higher the ratio of the long to short isoform, the faster the kinetics of aggregation. And what we know from recent studies, that the higher levels of soluble oligomers we have, it can cause in the end synaptic depression, so reduction in synaptic activity and reduction in brain activity, and in the end, memory decline. So as you can see, huge progress has been done during the last 20 years in biochemistry of Alzheimer's disease, but we still don't understand what is the cause of synaptic dysfunction. And to my opinion, there are several key questions that has to be resolved during last, I hope, 20 years. As I told you, we know a lot about familial Alzheimer's disease mutations. The main problem that these mutations happen only a few percent of Alzheimer's disease patients. However, how does experience, our everyday life experience, affect the ratio of these peptides and the level of the peptides in sporadic Alzheimer's disease? The most common form of disease remain unknown. The second question that we have to address, what are the primary neuronal changes that lead to synaptic depression? Okay? Because in many papers it has been assumed that synaptic depression is the cause, but it's still a question mark. And finally, of course, we have to understand molecular mechanisms that mediate these synaptic changes. And I will give you a few answers that has been done in my lab to some of these questions. First of all, how we address the studies. In order to really answer such a complex question, you have to investigate this brain system at different levels. Obviously, we start from nano level, and we use different biophysical methods to study protein-protein interactions, and we really invest a lot of time to do it in live system, in live neurons at the single synapse level, to really have a structure-function correlate of protein-protein interaction and synaptic function. Then we go into single synapse level. Synapse is around hundreds of nanometers, and we use different functional techniques to measure the activity of the synapses. And finally, we switch to electrophysiological techniques at the level of neuronal connections to study input-output relationships in these connections. And we also established microelectrode systems in vitro and currently in vivo to see how these changes affect whole neural network. And 4 years ago, we asked a simple question and say, if we ignore familial Alzheimer's disease mutations, right, because we don't want to work on something that represents only 1% of population, how we can address the question about the first and primary steps that can induce synaptic problems. And we decided to find a drug, and we found it, that reduce degradation of amyloid beta. So we just buy this drug that inhibits enzymes that called neprilysin, and when we inhibit the degradation or clearance of amyloid beta, we got very clean results. You can see this is functional imaging in hippocampal neurons, that after inhibition of degradation, there is a very strong increase in basal synaptic activity. So it's contradict completely right to the previous slide that I show you, that the primary deficits is synaptic depression. So then when we found these results, and we asked, first of all, whether it happened at any frequency, because you know, in our brain we have different frequencies of spike. We asked whether this action is frequency dependent, and the answer was yes. We found that if we inhibit amyloid beta degradation, it basically acts as a neuromodulator. It increases basal synaptic activity, and it does not affect synaptic transmission during high-frequency stimulation. So basically, it really shifts synapses from high-pass filter mode to low-pass filter mode. And after this, we asked if this is really the primary cause and how long it takes to synapses to get lost. We use the same drug, and we figure out that it takes at least 2 days to get synaptic loss. So you can look at this picture, and we counted functional synapses using FM dyes, and you can see that after 2 days of accumulation of amyloid beta, synaptic loss was prominent, but you also can figure out that the remaining synapses has been increased. What does this mean? We interpreted it as a homeostatic mechanism. So we lose our synapses, but in order to maintain brain activity, the remaining synapses are increased, but we still lose capability to react to high-frequency brain stimulation and to memorize. And we were very happy that several years after these papers, many laboratories in the world discovered that hyperactivity or increase in synaptic and brain activity was the primary change. And this is a work from Germany, from Arthur Kunert lab. And he discovered that in the hippocampus, first of all, he could find increase in calcium and synaptic activity, and only at the second stage, synaptic depression. So the second question that I promised you to address and to give you an answer, what could be experience-dependent mechanisms that regulate the ratio of long to short isoform, which is the main risk factor for Alzheimer's disease? So we asked how spike temporal patterns and mean rate, how properties of synapses, and how sensory experience could affect this ratio. And this has been done by really brilliant students in my lab, by Yiftach Dulev and Hila Fogel. And I will give you a brief answer. So this is the final answer of this work. So if you are looking at the ratio and on different frequencies of stimulations that we delivered, you can see that there is no change between very low frequencies of stimulation and amyloid beta 40 to 42 ratio. However, at high frequency stimulations, which are really relevant for learning and memory function, there is a significant increase in this ratio. And the main factor and the main change was due to changes in the short isoform, in amyloid beta 40 isoform. And this was completely contradictory to all the knowledge that we have until now. As I told you, familial Alzheimer's disease mutation affect the long isoform, and most of pharmaceutical companies are focused on this long isoform. But this data clearly says that the short isoform is the key determinant of activity-dependent dynamics in the brain. And finally, the main question was, what is the mechanism of these changes? So we used nanotechnology, and we asked very simple question. We say, let's see how this isoforms is produced. And these isoforms are produced by a very, very famous enzyme in the brain, and not only in the brain, by gamma secretase, and its catalytic subunit presenilin-1 or PS1. It has been known already that familial Alzheimer's disease mutation changed the conformation of this catalytic subunit. It brings it from open conformation to closed one. And the question that we really asked, whether spike burst or a high-frequency brain activity shift this conformation to the open one. To address this question, we use nanotechnology and phosphoresonance energy transfer technique, which really allows us to see conformational changes at the level from 2 to 8 nanometer, really at very, very short distances. And we took presenilin-1 and engineered the FRET construct. So basically, we inserted acceptor and donor at different parts of this construct. And we just asked, if we stimulate brain by single spikes or by high-frequency spike bursts, whether it can change conformation of presenilin-1. And the answer was that if we stimulate by single spikes, there was no change. But when we stimulated by spike burst, you can see that for 15 minutes, the conformation of the FRET efficiency was strongly reduced, and this was the answer. So spike bursts, which are really critical for learning and memory function, they produce conformational switch in presenilin-1 protein, and now we think about using this construct for early diagnosis of Alzheimer's disease. And finally, what I would like you to say, and what really is hopeful for us, because if you're looking many studies in Alzheimer's disease, there is no connection to the brain. Most of approaches, they are looking on neuronal cells, like a cell biology approach, like any other cell in the brain, in our body. But I always say that there is something different about neuronal cells, right? They can encode, store, and retrieve information which other cells cannot do. So we asked very simple question. If we give high frequency spike burst to different type of synapses, and we just use many pharmacological drugs. I don't have time to explain, but we used many different pharmacological drugs which are available in the market, and we change the properties of synapses. You can see this x-axis. You can affect the dynamics of amyloid beta 40 isoform, which is protective, right? And by this, you can affect the ratio of 40 to 42 isoform. So bottom line, our idea is that we can use combining two approaches. Using stimulating approach, there are many types of brain stimulations that can be used, and pharmacological approach to shift synapses to the high pass filter mode. And by this, we really hope we can delay Alzheimer's disease. So I would like to thank the people in the lab, mainly Eli and Eftach, and all the finding organizations, and I would like to thank you.